Dr. Matthew Hill, thank you for joining me.
Thanks for having me.
Where are you located? And can you tell people a little bit about your scientific background?
Right now, I am a professor in neuroscience at the University of Calgary at the Hotchkiss Brain Institute. I've been in science for about 20-ish years since I started grad school. Through that whole time, I've kind of always been focused on studying different aspects of the cannabinoid system in the brain, which is the biochemical target of THC from cannabis, studying the receptors, studying endocannabinoid molecules themselves, and kind of understanding how they modulate the way that the brain regulates things like feeding and anxiety and stress and kind of different homeostatic processes. More recently, moving into cannabis itself now because looking at some of the effects of cannabis on the brain and trying to understand that in a bit more depth.
Hmm. Interesting. You've done a lot of work on endocannabinoid biology, things related to stress, anxiety, pain, and there was a really interesting story a year or so ago, and you were involved in this, I believe, about a woman from Great Britain who had a really interesting and peculiar genome, and her endocannabinoid system was different from the average person's. So can you describe the story of that woman and what you guys discovered?
Yeah, so Joe Cameron kind of is this interesting, these are the kind of fun things in science that happens. Just this kind of random biological thing in one person informs us so much and creates this very interesting phenotype. And Joe, I mean, she was in her 70s, I believe. She kind of went through life not really noticing anything specific with the phenotype that she had for so long. It's actually kind of interesting. Apparently, the way that the doctors came across her is she had gone in for hip surgery, which is an incredibly invasive and typically painful process. And afterwards, when they had been offering her meds, she was like, "Oh, they gave her like one single Tylenol or something, and that was the extent. She did not want any more." And everyone was a little surprised by this, so they started asking her about, is this normal? And she kind of started describing how she's never really had pain. So then they started bringing in more people to have conversations and do tests with her, and they realized she really had this fascinating kind of inability to feel pain phenotype. And I mean, these people have been found before, but the way that their genetic mutations have been that have kind of made them not experience pain has been fundamentally different than what Joe had. More often, it's that there's some change in like a neurochemical receptor. These other people who are pain insensitive that impedes the body's ability to sense pain at all. With Joe,
Hmm.
it's unclear, I would say, if she can completely not sense pain entirely in some ways, as much as it just doesn't influence or affect her in any way. I mean, she describes kind of learning certain things like, oh, she'd put her hand on the stove and smell burning skin and be like, oh, don't do that,
Hmm.
which is a little surprising. But she did seem to have some sensitivity to cold still. So there
Hmm.
was some specificity in some of the things that Joe had. So I was first contacted by the doctors in Britain about this because they were really focused on the pain phenotype. This is a group that did a lot of molecular nociception work. And so they were curious about, they had started going through her genome, and what they had discovered was that she seemed to have this kind of mutation where she had a chunk of DNA kind of right around where the gene for FAAH, which is an enzyme that regulates the metabolism of endocannabinoids, right in that region, there was this kind of genetic effect, basically a lesion that seemed to take out a chunk, and it didn't directly affect the FAAH gene, but as a consequence, it seemed to influence expression of FAAH in some capacity. It's still not exactly clear how this is the case, but basically what it resulted in was that she seemed to have very little of this enzyme FAAH. And what FAAH does is it chews up and breaks down endocannabinoids. So if you have less of that enzyme, as a consequence, you'd predict that that means you have kind of elevated endocannabinoid tone.
Hmm.
And so you kind of like high endocannabinoids all the time. And this is an interesting thing to look at because of the fact that we make a lot of assumptions of what elevated endocannabinoid activity would kind of look like in a human. Is it going to look like what would happen from THC or not? Um, and so there have not been, I mean, we have some genetic variants in the endocannabinoid system that create small changes in endocannabinoid levels where they might be up a little bit. And there are phenotypes you can pull out, but nothing has ever looked like Joe Cameron. I mean, so they were asked me to measure her endocannabinoid levels. They sent me the blood. And that was when we found out that her endocannabinoid levels were quite high, higher than any human I'd ever measured, after about 1,000 samples from things we'd looked at before. And so it did seem like this genetic mutation that she possessed was somehow causing her FAAH to not function properly or be expressed. So that as a consequence, she was kind of living in this state of persistently elevated endocannabinoids.
Hmm. And so how much higher were levels? Was this 20% higher? Was this twice as high?
So as a frame of reference, I would say if I measure anandamide in the circulation of most average humans,
Mhm.
it falls between zero point 0.5 and 1 picomole per mil. So that's kind of the range you typically see it. People who have the more common genetic mutation in FAAH that doesn't completely cause it to have a loss of function, but it impairs it a bit, and that's been studied extensively. Their levels go up to maybe like 1.2 picomoles per mil. So again, not much of a change.
Mhm.
Joe's were about 2.2. So it was
Double or more.
more than double what the average person would have most of the time.
And so she had this interesting pain phenotype. She couldn't, it's not necessarily that she could not sense pain, but she certainly wasn't bothered by it in the way that the average person would be. Was anything else interesting about her, her personality or anything like that?
Yeah, so that was when they reached out to me, again, they were interested just purely in a pain context. And so after we had looked at the levels and were like, yeah, this person clearly has elevated endocannabinoids, I kind of went back to them and I said, I think it would be really interesting to look at what her emotional processing is like, simply because you would assume, based on all the stuff that we've seen with the animal work and what we've started to see in humans, that if you have elevated endocannabinoids, this should have a consequence on how you react to stress, how you process stress. So they started kind of doing interviews with her about emotional events. They tried to do some basic scales. She basically scored the lowest you can score in any kind of inventory of anxiety or depression. But interestingly, through the interview process, as they would talk to her, she would start talking about events that most people would have found like incredibly traumatic. Her first husband had committed suicide, he'd had bipolar, and she had lived with him through various periods that were very tumultuous. There had been a lot of other events. There'd been car accidents and stuff that she had been in. And the way she described these events was just very kind of like, this is shit that happens. Like, this is
Mhm.
life. Like, it wasn't, it just kind of rolled off her back. She didn't view it as being particularly salient emotionally in any way
Hmm.
necessarily. And she basically said she had never really experienced like the sensation of fear, really, or
Hmm.
being scared at stuff, which we found really fascinating. And I mean, prior to COVID, part of the hope was there would be a lot more investigation into this. And in fact, the colleagues in Sweden that I've been doing a lot of work with, looking at FAAH in humans, they became interested and they'd actually wanted to try and bring her over to Sweden to do some scanning and some more in-depth emotional batteries to try and get an understanding of her affective processing. But COVID happened, and so things kind of fell apart there, unfortunately. But it really is this like incredibly fascinating phenotype that she possesses.
Hmm.
With like a very highly sunny disposition and this kind of insensitivity to pain. It's largely what we would predict from someone who has a really high anandamide tone around the clock.
So anandamide is often referred to as the bliss molecule. And I used to wonder how true that was, just given, it sounds kind of like this pop science term that, a magazine would use to describe a molecule. So is that actually a pretty accurate description of this molecule in terms of what it does and what it regulates?
I mean, certainly some of the way that bliss molecule came out was because of how Rafi Mishulam named it. I mean, because
Mhm.
even in the word anandamide is the Sanskrit word for bliss, and Rafi's kind of inner joke was that this is like, the molecule of bliss in the body, based on how we conceptualize cannabinoids. Um, sh- yeah, I mean, I guess you could say to some degree. I mean, I always feel like it's difficult popularizing these things. I mean,
Mhm.
we went through decades of dealing with people using the term endorphin rush in an incredibly erroneous manner for virtually every description of stuff, because people learned that they got endorphins with something and they assumed that this meant everything was pleasurable. But I certainly, if from what we understand about what happens when you elevate anandamide, it does take the edge off of aversive events, and it seems to, I would assume, favor more positive salient events in the head, just simply by kind of reducing the salience of negative events. It would shift positive
Mhm.
bias towards things. And we know the endocannabinoid system tends to enhance rewarding aspects of stimuli. So I would assume, like, you know, based on the way Joe presents, she looks like she's just kind of happy-go-lucky with everything that happens to her, no matter how crazy things get. So in that sense, sure, I'd say it's got some validity to it, but there's always a downside. We just haven't explored it enough to see what it is yet.
So this woman has altered pain processing. She's in this persistent great mood, basically. What are like the downsides? Why isn't anandamide just at higher levels in all of us? Why did it not evolve that way?
Um, I mean, I think I'll take this from an anxiety perspective because that's,
Hmm.
what I study and what I think about. I would say, someone who has Joe's phenotype in an evolutionary context,
Hmm.
it's not actually a beneficial trait for a population to have because, and Joe commented on this herself. I mean, she's lived despite the kind of stressful and dramatic things that have happened to her locally with her marriage and other things. I mean, she's lived a relatively sheltered life in a small rural town in Scotland.
Hmm.
She hadn't been exposed, this wasn't someone who lived in Bosnia during the wars or was around during, Vietnam Wars or any kind of tumultuous period in recent modern history where
Hmm.
having a hypervigilant phenotype and being fully aware of the threats in your surroundings would be an advantageous phenotype to have to ensure survival. I mean, it's that kind of balance you would assume. When things are good, having a phenotype like Joe's is great because then you're not burdened by unnecessary anxiety or worries about things. But if you
Hmm.
live in a state or a period in history when things are highly tumultuous and there's a lot of threats in your environment, having that kind of casual, happy-go-lucky perspective is not going to ensure your survival very well because you are much greater likelihood to not recognize threats in your environment and put yourself at risk.
Hmm. So for
So
most of our evolutionary history, things haven't been as good as they are today for a lot of people, and we haven't lived in safe environments, and that's presumably why most humans aren't walking around with this type of phenotype.
yeah, I mean, I'd say anyone who I always kind of look at because of a stress person, you always look at things through an evolutionary lens, and you're like, there's a reason why evolution likes diversity in a population because things change so rapidly and things
Hmm.
change so violently at times that if you didn't have people sitting on all sides of a spectrum of any given trait at any given time with whatever the environmental demands are, right now Joe's phenotype is great, but yeah, 200 years ago, maybe not so much.
Hmm.
And so I think it's just evolution will always favor there being diversity because at different periods in time, there's going to be different traits that are advantageous and will ensure survival. I mean, we look at someone who has an anxiety disorder right now, who, like, if you're agoraphobic or you don't like going outside, you kind of stay in confines, or if you have social anxiety disorder and you don't like being around people as much, um, in our current, not the COVID environment, but our current modern environment, those kind of traits can be more detrimental to someone because of the necessity of social functioning and the way that things are played out. But historically, if you lived in a time of like a lot of threats and attacks, if you were someone who stayed at home and kept the kids there and didn't really want to leave because you were worried about what was outside, the chances of you surviving and your offspring surviving were a lot greater. So I mean, anxiety is crummy when it's out of context, but
Hmm.
it's actually an incredibly advantageous trait. So
Hmm.
I can see why, if we were all Joe Camerons, we would probably not have survived and thrived as long as we have.
So this was an interesting use case where someone has really high levels of anandamide. But can we back up a little bit and can you describe at a high level what is the endocannabinoid system and what is anandamide doing with respect to things like FAAH, this enzyme you mentioned, and things like the CB1 receptor under normal circumstances in most people?
So the endocannabinoid system is, I mean, it's just a very widely distributed biochemical system in the body. It's composed of enzyme or molecules and the enzymes that make those molecules and break down those molecules, and then it has the receptors that those molecules act on. The kind of thing that I think makes the endocannabinoid system interesting is just because of the biological kind of similarities and redundancy between the endocannabinoid molecules and some of the molecules that come from cannabis, the system was really discovered because it ended up being the biological target of THC from cannabis. And so THC binds to cannabinoid receptors. There are two that we know of and are well established: the CB1 receptor, which is really the one that's predominantly in the brain, and it's very widely expressed in the brain. It's very important for regulating neurotransmission and forms of plasticity. And then there's the CB2, which is primarily an immune receptor. It's mostly found on immune cells. It's certainly on microglia in the brain, might be on some neurons, but if it is, it's at incredibly low levels. THC acts on both of these, as do endocannabinoids. Um, the endocannabinoid molecules themselves, there's two. There's anandamide, which is one that we've been talking about already. And there's another one called 2-arachidonyl glycerol. They're again both very similar in structure. Um, They are regulated by entirely different enzymatic paths that make them and break them down. So the molecules do exist in kind of different worlds, let's say, because different processes will drive synthesis of one versus the other. The one thing I'd say the field hasn't entirely figured out yet is why there are two endocannabinoid molecules. I mean,
Mhm.
most neurochemical systems in the brain tend to diversify at the level of the receptor, where you have a molecule like serotonin or dopamine is a singular molecule and it signals through, 5 to 25 different receptors, depending on what transmitter system you're looking at. And that's where you see the diversification. Endocannabinoids kind of are the opposite because in the brain, we're really just talking about one receptor, CB1, that's just really widely expressed, but yet it's acted on by two different molecules. And if you look at the effects of those molecules, they generally do the same thing. They both activate CB1 in a similar manner to how THC does.
Mhm.
The CB1 receptors kind of got this interesting localization because it's found almost exclusively on axon terminals. So what that would mean is when it becomes activated, what it basically does is it turns off neurotransmission because it's an inhibitory receptor. So whether it's activated by THC or endocannabinoids, when CB1 becomes active, you just see less synaptic neurotransmitter release. That doesn't necessarily mean it's a depressant, like some drugs are kind of central nervous system depressants, because if cannabinoids are turning off inhibitory transmission in the brain, they're actually enhancing excitation. So it's very
Mhm.
synapse specific and can get incredibly complicated with how they actually regulate brain activity when you kind of look at the net effects together. But I mean, what anandamide or 2-AG would kind of normally be doing is, I mean, they certainly are involved in regulating basic processes and making sure that the brain exists in, I would say a homeostatic range. Like if you really had to boil down what the point of endocannabinoids are in the brain,
Mhm.
the main argument I would make, and I think a lot of people in the field would make, is they're really there to make sure everything kind of keeps in check. I view them in many ways as a molecule of homeostasis. Like
Mhm.
their main job is to keep things where they should be because of their ability to regulate neurotransmitter release. If we look at something like, epilepsy, where you have a state of hyperactivity and there's too much excitatory transmitter being dumped out, endocannabinoids would be trying to curb that and put the brakes on the system by activating CB1 and limiting the amount of glutamate release. So they're trying to prevent that hyperexcitation and overexcitability of a circuit. So that's, I mean, they're really a buffer in many ways to make sure that the brain's activity doesn't go too far to control one way or too far like down in the other direction. So they can really gauge the kind of basal activity of a circuit and respond appropriately to make sure that parts of the brain behave the way they should. But if you
Mhm.
take a kind of bigger step up at a process level, like something like feeding, which you're very familiar with, but like, endocannabinoids are a really important regulator of feeding processes. So we know that like if you go a period of fasting and you don't eat, or an organism doesn't eat for a period of time, and obviously start burning through your energy reserves in the body, and so your body wants you to eat, endocannabinoid levels seem to start elevating within feeding circuits, like in parts of the hypothalamus. You see endocannabinoid levels go up during periods of fasting, and they kind of shift the excitability of the circuits to try to enhance the activity of clusters of cells that are going to drive feeding activity and enhance them. So they almost act as this on switch to try and drive feeding behavior. And then once food consumption occurs and macronutrients hit the body, even as quickly as when the gut starts sensing them, endocannabinoid levels in the hypothalamus start plummeting and going back down. And so they're very responsive to nutritional status and hunger state. And so in that sense, again, it's like a homeostatic mechanism. You haven't eaten long enough, your body needs to eat endocannabinoids, mobilize in a process to drive you for food seeking behavior. And then once you've eaten and satiated yourself, they go back down. In the sense of stress, we see the same thing. Endocannabinoids seem to, at rest, be kind of keeping some circuits in the brain, especially within the amygdala, kind of quiet under ambient conditions when there's no threat. And then what we see is that in response to a threat, there's this really rapid loss of an endocannabinoid signal, and that seems to facilitate activation of these circuits that drive like the generation of an anxiety state. But then as we go into the stress response, we mobilize hormones like glucocorticoids, like cortisol in humans, and these hormones then seem to actually jack up endocannabinoids in the brain as a means to turn everything back off and help turn off the stress response and help us come back down to a resting state so we're not kind of hyper vigilant and anxious still. So again, like in almost every process you look at, you can make the same argument for pain. I could say the same thing for inflammation. You see the same thing over and over with how endocannabinoids behave. They really respond to disturbance in a physiological process. They act to modulate a neural circuit to influence a behavioral drive that will help bring things back to where they should be. And then once everything goes back to normal, the levels start to normalize and the signaling goes back to this kind of low level tonic state where it keeps things, at rest.
Mhm. And is this why endocannabinoids are so localized in space and time? They tend to get used when and where they're needed. They're not just sort of flooding the entire brain or anything like that.
Yeah, yeah, I think it's something that a lot of people don't appreciate is how precise that kind of spatiotemporal action of endocannabinoids is. I mean, like when a neuron releases it, it only hits a couple of synapses, and it's very specific in how it influences those synapses. So, I mean, and this is always one of the reasons when people ask, like, if you have a drug that will boost endocannabinoid levels, like a FAAH inhibitor, where you inhibit anandamide metabolism and anandamide levels go up, they're like, why doesn't that like look the same as when someone smokes cannabis or consumes cannabis and THC hits the brain? And the only thing you can try and say is, well, you got to think like anandamide's not being made everywhere in the brain where there's CB1 receptors, presumably, because some of those are going to be more hit by 2-AG,
Hmm.
and the amount of anandamide that's being made by a cell is going to be, if it's nothing driving it, it won't be that high. So if you inhibit FAAH, anandamide will start accumulating, but it's only going to be accumulating in the parts of the brain where it's normally active.
Hmm.
If you consume cannabis and THC gets into your bloodstream and hits the brain, it indiscriminately will just saturate every CB1 receptor it can find. And so that seems to be one of the main reasons I would say you get this very big difference between boosting endocannabinoid levels and actually consuming cannabis in terms of the psychological and intoxicating effects you see, because THC just has no discrimination. It hits everything it
Hmm.
can because it's not dictated by synthesis the way that endocannabinoids are.
And is this so when we think about the normal function of the endocannabinoid system, you've essentially told us it's this homeostatic mechanism that regulates more or less everything from, pain and anxiety to feeding behavior to all sorts of stuff. Is that why something like cannabis, something with THC has so many different effects and side effects that seem to go in every direction? Some people say that they consume cannabis, it makes them less anxious. Some people consume cannabis, it makes them more anxious. Does it have to do with this flooding of the brain with THC and the fact that these receptors are controlling so many processes?
Yeah, I think I mean, the joke we always have is like cannabinoids influence everything but regulate nothing because they're not essential for anything to occur. They modulate everything. Um, but I think for sure, I mean, that's why there's so many diverse effects of cannabis on someone when they consume it. I mean, you have hemodynamic effects on blood pressure and vasodilation. You have, some effects on feeding, which again, like you said, can go in either direction. I mean, we classically think of the munchies as a consequence of cannabis, but you consume too much THC and it goes in the complete opposite direction. People can start vomiting uncontrollably.
Hmm.
People lose their appetite if they kind of, I don't want to say overdose, but overconsume THC or cannabis and they have too much of it. Like you said, most people, most recreational cannabis users use cannabis because they say it reduces their stress and anxiety. And if you ask people who've tried cannabis who do not use it recreationally, almost always they say because it made them anxious.
Hmm.
So it's interesting that you get this. And I always say it's weird. It's not like a graded response where some people have reduced anxiety and other people it doesn't really do as much. It's like a polar opposite response where for some people they find it helps them calm down and other people it throws them into a panic attack. So it's very interesting that a drug can do that when it's one drug targeting one receptor. But some of this is going to be individual variation with receptor expression. And one of the things that we've learned from the animal side of things is these kind of opposite effects, like what you were mentioning with anxiety.
Hmm.
This seems to be dictated by CB1 receptors on different neuron populations.
Hmm.
So what we've learned from mouse genetics is that if we remove cannabinoid receptors off of excitatory neurons, so cannabinoids can no longer suppress excitatory neurotransmission, we lose all the effects of THC in terms of the standard intoxication effects.
Hmm.
So like sedation, the changes in the body temperature, pain processing, all these things, they're all lost if you take CB1 off of excitatory neurons. If you take it off inhibitory neurons, it does very little.
Hmm.
Yeah, all the effects of THC look pretty similar. However, if you start going to really high doses of THC where you get these opposite effects, then that seems to be where CB1 on the inhibitory neurons plays a role. And so, in the context of anxiety, because that's the easiest one to explain, you can take this to the amygdala. So, if we look at the amygdala in the basic sense of we know the amygdala becomes too excitable in anxiety states, and in individuals who have anxiety disorders, it's hyperactive. There's more activity than we should normally be having. And we know things like benzodiazepines and most drugs that treat anxiety reduce neural activity in the amygdala, usually by promoting inhibitory transmission. So if we look at how cannabinoids regulate anxiety, what we've learned is that in the amygdala, if THC activates CB1 on an excitatory neuron, it decreases the amount of excitatory transmitter like glutamate that's going to be released. And
Hmm.
as a consequence, in the amygdala, that will calm the amygdala down.
Hmm.
And so that seems to be why THC at moderate and lower doses is reliably anxiolytic, because it seems to be primarily suppressing glutamate release in the amygdala and keeping it quiet. As you start jacking doses up and you go to a higher level, then it starts to saturate CB1 on GABA, and it seems to do the opposite, because now what it's doing is turning off inhibition, and then you get the opposite
The
effect where you
opposite
start
effect.
getting more excitation. And so you get this, it's almost like you kind of cross a threshold and it flips.
Yeah.
And suddenly, before it was quieting it down, and now it's making it more active.
So that's interesting because in the scientific world, you often talk about something like an inverted U shaped effect where something switches at a low versus high dose. And that's what this sounds like in the cannabis world, where people aren't really so familiar with some of the mechanisms that can be at play here. People talk about a multiphasic or a biphasic effect.
Yeah.
And it sounds like that's exactly what we're talking about. And the basis for that is at a relatively low dose, you're affecting one type of neuron in the brain, say an excitatory neuron in this case. An endocannabinoid at that dose would actually make things quieter. But then you go up higher and you start hitting those inhibitory neurons and you get the opposite thing happening.
Yeah. Yeah, and that's, I mean, so if you delete CB1 on glutamate and you give them low dose THC, it doesn't reduce anxiety anymore. So you need it there. And exactly like you're saying, if you delete it off of the inhibitory GABA neurons and you go that high dose where they normally get anxious, they no longer get anxious. So we
Hmm.
know that those different receptor populations mediate it. But you're exactly right. It is entirely an inverted U curve. And normally where we would sit is probably somewhere on the ascending arm of the first part of the U,
Hmm.
because depending on what your basal level of endocannabinoid function is, because we know if you completely disrupt endocannabinoid function, that also enhances anxiety. But we know if you kind of go all the way around the inverted U and you end on the other side, too much also increases anxiety. But the disrupting, if you reduce endocannabinoid function, that is probably increasing anxiety, because now you're affecting the CB1 and glutamate. So again, you're causing more excitation
Hmm.
because you're kind of removing that brake system that would normally exist on the excitatory neuron. So it is entirely an inverted U curve. But yeah, biphasic tends to be how people in general public will refer to it. That you're usually safer with low doses, and you go higher doses, and everyone has a different threshold, but you cross that threshold, and kind of everyone will have an adverse event.
Yeah, yeah, I definitely know people who, I mean, this all makes a lot of sense because, I know people who can consume a lot of THC and have
Mhm.
no side effects at
Yeah.
anything approaching a normal dose. And then there's other people who even 2.5 milligrams of THC can be problematic.
Yeah, and the thing is, I mean, there hasn't been enough work in this, but it's interesting because it makes you wonder how much of this is just dictated by individual variability in CB1 expression.
Hmm.
So one of the things I've always been fascinated by is there's a lot of reports in the literature that women will have a kind of a narrow window to have an adverse event. Like women will more likely have an anxiety or a panic-like response if they consume cannabis, even at lower doses than is typically seen in men. And some of the stuff we've seen in the animal work and some of the suggestions we've seen come out of the PET imaging in humans is women may indeed actually have more CB1 receptors than men, which just means that window of when you go from too little to too much might be a much narrower threshold. Now, if this was the 70s, the 80s, when cannabis was like 5% THC, you would have a lot more room to play with that window. But
Yeah, yeah.
nowadays, when you're talking like 25% THC, this can, like, you could probably cross that threshold with like a single toke. Like
Hmm.
one toke is not going to do too much, two tokes is too much. Like that's
Hmm.
how narrow it seems to be for some people from what I've seen and heard about like how quickly they can go from it being okay to not being okay.
Yeah, I want to talk more about this concept of the endocannabinoid system acting as a stress buffer. You've so far described it as almost like a universal buffer. It's regulating almost any process you can think of in a homeostatic fashion. So can you maybe back into this by describing a typical like healthy stress response? What's happening in the brain and body under a state of stress?
I mean, normally what will happen is, you kind of think you're just going about your day, things are fine, there's nothing there. And then, like the example I tend to use, just because I get something people can get to, is like, say you're walking through a forest and you're just kind of whatever, you're on a hike, you're doing your own thing. And then all of a sudden you hear like a stick break. And it's, there's nothing in your vision that you can see, but immediately everything about you changes. So you've suddenly, what has happened is your brain has perceived that there's a potential threat in your environment. And this is basically what will be the most classic trigger for a stress response is your brain perceiving an immediate threat around you. And what happens very rapidly is you'll get a dump of norepinephrine into your forebrain, which causes a lot of shifting and reorientation of your attention so that you start to focus on details in your environment that you probably were taking for granted prior to that. So this is the shift into a state of hypervigilance where, like, if you're in the dark, what you'll notice is your pupils will rapidly dilate and you can suddenly see a
Hmm.
lot more than you could before because now more light gets in because your body is trying to prepare you to look around and figure out where that threat is coming from. The adrenaline, when it hits your bloodstream, aside from your pupils, will make your heart rate go up, makes your blood pressure go up, so that you are ready to deal with this threat as it presents itself, whether it be running away or engaging in a fight. Um, and you kind of, your brain also, in terms of anxiety, especially if you can't see where the threat is, your brain is now constantly trying to search and figure out where that threat is coming from so you can assess how potentially damaging it's going to be for you and whether this is going to be something you're going to have to contend with or not. Because maybe it was just a stick falling, we don't know. So that kind of way that your body flips into that mode prepares you to deal with the threat at hand. The other thing that happens is you get the kind of hormonal response, which is this release of cortisol. The main function of what cortisol does is when it hits your blood is it causes a mobilization of blood glucose, essentially. So the way I always try and explain this to people is, in like the classic form of the stress response, is the adrenaline component is basically the mechanism to get the trafficking of oxygen and glucose accelerated so you can deliver this to your muscles and your brains so you can run away and you can focus on your environment. And the cortisol arm of that response is basically giving you more fuel. So
Hmm.
it's basically like, okay, let's get more sugar into the blood, let's make sure that we have glucose to feed the brain and to feed the muscles so that we don't fatigue. And you kind of go into that state. Now normally you should be able to assess whether there is a threat within a couple of minutes. So the stress response in a typical sense is very short-lived. It's really an assessment. You've got to figure out what's around you. And once you've assessed that or removed yourself from the situation so the threat is no longer present, everything has to kind of turn back off. I mean, the adrenaline response is obviously very quick. It's very quick to turn on. It's very quick to turn off. The cortisol response, again, is a little bit more slow. But one of the things I think that we hadn't always appreciated in the brain, let's say, is like when I started doing work, the impression I always got from the way the field described stress recovery is that it was essentially a passive process. That once the brain had said, okay, this threat isn't present anymore, um, no need to keep driving the stress response. It just basically turned the light switch off. And then what you saw was like a passive recovery phase. And actually, I don't think that's the case anymore. And I think what we've really learned about the endocannabinoid system in this context is that it is an active mode of stress recovery. So it is becoming mobilized by the release of these stress hormones. And part of what it's doing in that state then is to turn the amygdala back off, because the amygdala becomes very active in response to threat. It's what kind of helps shift our perception to try and do threat detection in our environment. So as we calm the amygdala back down as endocannabinoid levels go up, this is part of the normative recovery response. And one of the things that, you think about this in the context of how this could be pathological is it's like not everyone turns their stress response off properly. There are
Hmm.
people we can see who stay in that, they get pushed over the edge to have a stress response and they kind of stay locked in it for some time. I mean, there are also people on the other end of the spectrum, like who would load high in psychopathy, who never mount a stress response because they don't really perceive anything as a threat. So, you can kind of see pathologies on both sides of the spectrum there of how not mounting a normal stress response, um, if it's too much or too little, why it doesn't, effectively work in the long run.
Hmm. So I start to think about things like PTSD here, where you have some kind of stress or anxiety response that fails to shut off properly. And based on what you've told us, the endocannabinoid system is at least an important part of the equation for the switching, the turning on and the turning off part. So, can you maybe talk about, what is PTSD from a scientific perspective, and what do we know about why some people have this dysregulated stress response that outlasts the actual threat?
Yeah, I mean, PTSD is I mean, it's not as complicated, let's say, as something like depression, which is super, super heterogeneous. But PTSD it's very, I don't know. it's hard to describe in many ways. In terms of why some people have this response after exposure to a traumatic stressor and others don't, I wouldn't say the field has a strong grasp on that.
Hmm.
I would say that's kind of like a big thing that people have been trying to understand for some time. Is there some way we can predict who, if 10 people get exposed to a trauma, which one of them will subsequently develop PTSD and who wouldn't? That obviously would be incredibly valuable information for us to understand. It seems more likely that it's probably some latent biological signal that doesn't present itself until the trauma itself has actually unmasked this
Hmm.
latent difference that we're seeing, and that has made it very difficult. What we do know we see is in the aftermath of the trauma, there are kind of various trajectories that people will have. There's kind of the super resilient phenotype where there's no short-term effects, like there's no kind of acute stress syndrome right afterwards, but also there's no development of PTSD. Then there's the kind of group of people that do have this very robust acute stress syndrome in the immediate aftermath of the trauma, but it kind of wanes as if a normal stress response over
Hmm.
time and it goes back to normal and there's kind of no long-term changes. And then there's I think what a lot of people think is more classic PTSD, which is in the immediate aftermath of trauma, there's not really any visible traumatic stress symptoms. And then in the weeks and months after the traumatic event, things begin to emerge and present themselves. And then you have the last, which is just the people who go right into the traumatic state and stay there, and it goes into
Hmm.
full-blown PTSD. What differentiates individuals that we look at post hoc? There does seem to be, alterations in the way that the brain is processing information. There seems to be some issues with how, say, the prefrontal cortex is communicating with the amygdala. So
Hmm.
the prefrontal cortex should, in many ways, kind of keep top-down control over the amygdala and prevent it from flipping into states of hyperactivity when there isn't threat. Like, because the prefrontal cortex is so important for reference memory, a lot of the thought is that there's this conversation. I hate anthropomorphizing how brain structures talk to each other, but it's kind of the easiest way. But there's kind of this conversation where events present themselves and the prefrontal cortex is kind of telling the amygdala, no, this is safe, don't worry about it, we've seen this before, or this isn't a threat anymore, it was before, so now it's
Hmm.
time to calm down. So if the prefrontal cortex isn't having that conversation with the amygdala the way it should be, the consequence of that in some ways is that the amygdala is going to kind of go out on its own. And the default mode would often be threat detection, to say, okay, from a survival perspective, if you don't have that input telling you what's safe and not safe, from a survival perspective, the default would be everything is potentially dangerous, so act accordingly. And so the lack of functional connectivity that we seem to see in PTSD would suggest that you're not having that top-down control over the amygdala as much, and that may be influencing certain aspects of this. There's certainly states of hyperarousal and hyperautonomic function. So individuals with PTSD definitely will have elevated adrenaline. They will go into episodes where they'll have kind of re-experiencing events and panic where adrenaline levels will shoot up and they'll have massive autonomic changes. And the sleep, their sleep patterns are often really, really messed up, especially REM sleep can
Hmm.
be kind of this period where they start having very violent, traumatic, re-experiencing nightmares. And there is a lot of belief now that actually that sleep phase is very important for kind of reconsolidation of the traumatic memory and the way that it evolves and generalizes over time.
Hmm.
And that this is due to some kind of disruption in REM sleep that's favoring this re-experiencing. And then because of that, there's also this horrible quality of sleep, which obviously
Hmm.
contributes to a lot of the other issues.
I think an interesting thing to talk about here are the different potential treatments that different people are exploring and advocating for PTSD. So staying on the notion of like sleep and nightmares first, there's there's no hard evidence for this, but I believe there's indirect evidence that THC and cannabis that contains THC might suppress REM sleep. And some people have attempted to at least make the connection that perhaps medical cannabis could be useful in treating PTSD. The idea being if it is suppressing REM sleep, you take it to help you fall asleep, you're suppressing these nightmares, and maybe there's something there. How much is there that we know
Yeah.
about in terms of medical cannabis?
So it hasn't, it sadly, it hasn't been done as much depth as it should. So most of, I mean, you're right in everything you said. So, I mean, there is some literature looking at, a lot of this was done in the 70s when, you could do this way better now with sleep architecture studies, but It's certainly, there is evidence to suggest that when you give someone THC, you do get a suppression of REM sleep. I mean, even in the initial FAAH-inhibitor trials, they saw this. They saw that it reduced REM and it favored non-REM sleep, which is interesting because it does tend to also increase total time sleeping. So not only are you getting sleep, but it seems to be favoring more like, I guess, light sleep and deep sleep phases over REM,
Hmm.
which could have some benefit for people, but definitely in the context of PTSD. So I agree. I've always thought that would make sense. And the reason I think that it makes a lot more sense is, if you look at the evidence that's actually been done in populations of people who are afflicted with PTSD, there's kind of clusters of anecdotal studies, which, you would usually normally not put a lot of weight in. 1,000 anecdotes are still just 1,000 anecdotes. It's not data. But if you're a good scientist in my mind, if you have 1,000 anecdotes that all say the same thing, then that is where you form a hypothesis from and design a study to test it, because you would
Hmm.
be stupid to not pursue that. So then people started looking at this as kind of retrospective things in clinical populations, where they looked at what were people saying when they got put on THC or cannabis. Were people with PTSD claiming now that they were having less nightmares or improved sleep? And this kind of popped up again and again in these chart review studies that were really not well controlled. So in terms of actual science, there's been two studies that were done, and both of them were done by the Canadian military, and they were done using an isolated THC pill. It's Nabalone. It's like a synthetic THC that's pharmaceutical grade. And in the first study, they just did an open label trial. So this was, again, not properly done necessarily because it wasn't blinded, but were dealing with a population of war vets that all had treatment resistant PTSD that were very bad with nightmares. So people who had incredibly bad nightmares and very poor quality sleep. And they hadn't responded to propranolol or any of the benzos or any previous drug treatments that had been attempted on them. So they gave them Nabalone and just tracked them. So it
Hmm.
wasn't blinded, but what they found was a very profound effect. I think it was something like 85% of people on the first night stopped having nightmares.
Hmm.
And they tracked it for a while, and it looked like it. As soon as they took them off it, the nightmares reemerged and came back. So it seemed like, okay, that was some decent evidence. So from there, they actually did do a double-blind, placebo-controlled crossover design study, which is what you should do. The only downside is it was kind of small. It was only like 12 or 15 subjects, so it wasn't huge, but at least it was done properly.
Hmm.
And this study basically built off of what they had found and replicated it even more substantially, because in the crossover arm, it was very clear that regardless of whether they knew they were on it, like because they're blinded, as soon as they were taken off of the THC and put onto the placebo, almost immediately the nightmares came back and
Yeah, and you would
sleep quality
expect that.
plummeted. Yeah.
I think you would expect that, right? Anything from, this study to SSRIs, there's this phenomenon of REM rebound, which I know is quite robust. If you suppress REM sleep and then you stop suppressing it, it actually comes back with quite a force.
Yeah, I mean, this is certainly something you see in cannabis withdrawal studies. When people go off of cannabis, when they're trying to go into cessation, they often will report like very intense nightmares over the first few nights.
Hmm.
It's obviously different than what's going on in PTSD, but yeah, I mean, and the thing I take from that as well is it's not only like, are you dealing with a REM rebound, but like it's not a cure. It's a treatment. Like it would
Yeah.
be, if it proves to actually be effective in the sense, this is like a real-time management tool. This is not, I mean, I think that's where some people get confused by things. They think that, oh, it means it cures something. It's no, this doesn't, because as soon as you go off of it, it comes back. So this is really just a tool to help manage a symptom like this. But it seems to be an incredibly effective tool in the sense that if this works, the question is, can this sustain over time, or will this develop tolerance? Will there be other consequences? I mean, obviously, some of that would be influenced by if someone starts escalating use over
Hmm.
time versus, a lot of the ones in Canada, at least in the military, when they interview them, they say that they only use a small amount of cannabis and they only use it at night right before they go to bed. And they've been doing this for years and they've not developed tolerance, and they
Hmm.
will report consistent benefit on their part. But again, this hasn't been studied properly, so it's really hard to say definitively.
Interesting. So, assuming the results from Canada held up and are robust, it is something, it is a tool, it's a management tool, but it's immediately effective. That would be the upside.
Yeah.
The downside is, we haven't done the studies. We don't know if it's going to be effective forever, and it's not a cure, it's a management tool. There's two other treatments based on your work and other people's work that come to mind in terms of treating these things. One would be FAAH-inhibitors, and I don't know much about the clinical work there, so I'd love if you could describe that. And the other one that's probably the most famous right now are the MDMA assisted psychotherapy trials, which do
Hmm.
seem to be a potential cure. So can you kind of
Yeah.
compare and contrast each of these tools that we've been researching?
Well, I'll start with MDMA just because I can't really say much about it. I mean,
Hmm.
I've seen the clinical data. The psychedelic stuff is interesting. I mean, there's some early stuff with psilocybin as well. Um, I don't, I mean, to me, this is more than just simply a biological mechanism. This is like, there's something about the experience. The hallucinogens are creating an altered state of conscious that are allowing people to either have some kind of differential impact from therapy or consolidate that information in a different manner so it's more sustained, or it allows them to access the core trauma in a different manner that they can psychologically process it. I'm not sure what's going on there. It does seem to work with the right therapist.
Hmm.
So I'm very curious to see where the psychedelic space is going to go as now there's more recognition of this. And so there's kind of increasing resources being put into it to kind of study it. So as for the FAAH inhibitor side, so I definitely think the FAAH inhibitor side has a lot of potential. This is something I'm very excited about to see that it's finally moving forward.
So
Um,
just for background, so you talked about FAAH in the beginning, but when you say FAAH inhibitor, you're talking about a drug, so a pill
yeah.
that you would take, a pharmaceutical. It would inhibit this enzyme called FAAH and thereby increase anandamide levels.
Yeah, yeah. So you get a nice sustained increase in anandamide levels. They go up quite a bit, like even higher than you'd see with Joe Cameron.
Oh wow.
But they're going to those levels. Um, and so so we've done, like I was involved in one trial that was done in Sweden. We helped process some of their blood samples to look at some of the outcome measures for the anandamide effect. It was just a study done in healthy controls. And so this was Marcus Heilig and Leah Mayo. And what they did was they just dosed people either with placebo or a FAAH inhibitor for 10 days. So you had nice stable drug levels and a nice stable sustained elevation in anandamide. And then they did a couple of things. They put them through kind of a stress challenge where, you know, you have to do this public speaking and mental or verbal arithmetic stuff in front of a crowd. And it kind of really stresses people out and you get a nice solid stress response in people. And they also did an acute kind of fear conditioning and fear extinction task where, you know, they try and couple a stimulus with an aversive outcome. You get a nice stable conditioned response and then you try and extinguish it. And I'll just quickly kind of sidestep to say that the fear extinction thing is really important in the context of something like PTSD,
Hmm.
because seems to be one of these processes that is not working normally in PTSD. They can't extinguish aversive memories the
Hmm.
same way that normally can. So
And,
this is
well,
one of the reasons why there's been this interest in endocannabinoids there
y-
too.
yeah, and for people who don't know, fear extinction, that would be the process by which the brain learns to stop responding to something that is no longer a threat.
Yeah. So the way I would say this is it's, you know, if you had been in a situation where you'd been, let's say you're in Vietnam War and you'd been in the forests, and if you heard a bang when you're in the forest, you're like, you know that that's predictive of an aversive outcome. You know, they saw this in a lot of the Vietnam vets. They came back, they would, you know, go for a hike and you'd hear something and you would respond as if you were still in Vietnam because you're in a forest and you hear a bang.
Hmm.
Now, the extinction process is understanding that this is a different context now. So even if I'm in a forest and I hear a bang, here it's not going to relate to the same dangerous outcome as it did when I was in Vietnam. That's kind of the way that you'd uncouple those processes. Or even something like a stove, if you put your hand on a stove and you burn it, you're like, oh, you immediately instinctively remove your hand away from it. But you'll learn over time that if that element isn't turned on, it's still safe to put your
Hmm.
hand down there, so that you're kind of learning that the stove isn't always dangerous. Like it's a specificity thing. So if you can't fear extinguish, you just kind of become permanently scared by the stove, or you'd always be terrified in any forest
Hmm.
if you hear a bang. So that's kind of if it doesn't work right, what that process looks like. And what the animal literature would say is that cannabinoids can augment or accelerate fear extinction and make it occur more readily. So people will learn faster that something is safe, or they'll learn faster that it's not dangerous anymore. So that was kind of what Leah and Marcus were wanting to test in their study, was in humans, does this look the same? And so what we saw from the trial after giving 10 days of this FAAH inhibitor, so if we look at stress, yes, the FAAH inhibitor blunted the kind of adrenaline response to stress. So if we look at the sympathetic response, looking at skin conductance or another readout of adrenaline, that was blunted if you had high levels of anandamide. So that's consistent with what we would expect from like what we know about the stress response. They also did some unbiased measurements of facial muscle changes. So, you know, our facial muscles can become very expressive. And like one of the things that we tend to do is if something's negative, we'll like wrinkle or furrow our brow, versus if something's positive, you smile. So you can measure different muscle tone on the face. And what they found was that normally when you get stressed, there's more reactivity of the muscles that are like indicative of like a negative facial expression.
Hmm.
And the FAAH inhibitor selectively blunted the ability of stress to promote those muscle changes as well, which is just another kind of unbiased readout. And even when they asked them, how stressed are you? Those that had had the FAAH inhibitor self-reported less stress responses. They felt less stress. So it kind of recapitulated what we would have expected. And on the fear side of things, it's interesting because one of the things that we've learned from the animal stuff that appears to be true in humans is it doesn't do elevating anandamide doesn't do anything to the fear conditioning process itself. And
Hmm.
that's actually really important because
Yeah,
we
yeah.
need to be able to couple aversive events for survival. So the animal work has shown it doesn't really do much there. Now in humans, we know their fear conditioning looked entirely normal. But then when you look the next day, when you go through fear extinction, they seem to fear extinguish better and they remember it. So if you look at how well the consolidation of that fear extinction memory was, the FAAH inhibitor augmented that quite a bit. So that was kind of really promising because it was the suggestion that, we are replicating a lot of these anti-stress and kind of pro-fear extinction effects that we've seen in animal work, and what we would assume and what we'd be wanting to look for as hallmarks of potential treatments for something like PTSD. Um, so that was very encouraging. And then more recently, there was a study that just came out from Johnson and Johnson with their FAAH inhibitor, and it was in the context of social anxiety disorder. And what they were able to show was, again, somewhat we predicted. They had a bit of dosing issues. It looked like the dose that they used was kind of right on the cusp of being able to effectively elevate anandamide. Their anandamide levels weren't super high. And so I think they're redoing this now. But when they looked at the individuals that actually had the elevated anandamide from the FAAH inhibitor, they did find that it had benefit in improving symptoms of social anxiety disorder. So it did kind of seem like it was doing what we would expect in that sense. And then almost immediately after that paper came out, another paper from Johnson and Johnson in humans using that FAAH inhibitor came out looking at amygdala reactivity and also showed in humans just by simply dosing people healthy controls with the FAAH inhibitor and then using threat cues to activate the amygdala, we could blunt that, or they could blunt that at least by elevating anandamide. And the only other real clinical thing that's been interesting that's come out is the cannabis withdrawal studies that have been done, where they've been showing that individuals who are trying to quit using cannabis, if you give them a FAAH inhibitor, then they are able to reduce their cannabis use. And it also, in that context, improved sleep and anxiety. Sorry, this piece of hair. I'm just going to try and get this down. I don't know why it's poking up, but it's driving me insane. Okay.
So, okay, so in summary, the FAAH inhibitors look pretty promising. They're not messing with fear learning, but they are aiding fear extinction, which is exactly what you would want for something like PTSD. And I don't know the answer to this, but I would speculate that the FAAH inhibitors might not have that many side effects. So what do we know about any negative side effects?
Yeah, so it's funny, the way that I always had this described to me from the pharma side of things was they said FAAH inhibitors have the side effect profile of a glass of water, which surprisingly I learned is actually a concern from a pharmaceutical perspective, because the concern is that means it's not having enough of a biological impact, because
Mm,
everything, like there's always some side effects, even things like aspirin, like every
mhm,
over-the-counter drugs. And so they were actually initially concerned that the FAAH inhibitor wasn't even getting in the brain because they couldn't see any, like there was no psychoactive effects, there was no observable changes, no one could report it. Like people
mhm.
who'd been dosed couldn't tell if they were on drug or placebo. So they
Wow.
actually did a PET imaging study in the brain to make sure it was getting in, and it was. And then they did the REM sleep study, and that's how they learned that, okay, there was a central signal, because they could get a suppression of REM sleep by giving FAAH inhibitor. So it really doesn't seem to. I mean, there is some confusion out there because there was that disastrous trial that happened in France with a FAAH inhibitor where someone ended up dying. But that, as we learned in the aftermath, actually had nothing to do with the target of FAAH, but it had to do with really bad chemistry on the compound itself.
Mm,
And it seemed to be forming covalent bonds with like liver enzymes and preventing drug metabolism. And doing a multi ascending dose trial did not proved to be a safe way to go with that without having done the proper talks in advance.
mhm.
But as an actual target, with all the data that's come out of, Pfizer's had one, Sanofi's had one, Merck's had one, Johnson Johnson's had one. These are multiple companies that have gone through phase 1, some into phase 2. None of them have ever reported anything significant as a side effect profile, which is actually very encouraging.
Mhm. And so is that where we're at in the clinical process? We're not at phase 3 yet, but maybe about to start that?
Yeah, I would say there's still a lot of, there was such a I mean, the FAAH inhibitor, when it first got made by Pfizer, it unfortunately was first used in a very shitty kind of pain trial that didn't work.
Hmm.
And that just immediately deterred everyone from going near FAAH inhibitors. And I'd say it
Mm.
killed the whole FAAH inhibitor program for at least 5 years. It was very frustrating because they had put all this work into developing a really, really clean drug. Um, and then so they started to get some interest in it again, but the problem was once interest in the FAAH inhibitor started again was right around the time pharmaceutical companies kind of decided to abandon psychiatric and neurological diseases, which has become a trend because,
Just
I
too
mean,
hard.
yeah, and as you and many people know, there hasn't really been new classes of psychotherapeutics for psychiatric or neurological drugs in decades. I mean, we're
Hmm.
basically repurposing old mechanisms and trying to create new flavors of the same drug. FAAH inhibitors actually represent an entirely novel target that has never existed before from a drug perspective. So that's why I think it's actually kind of exciting.
Hmm.
Um, but I think the problem was like when the pharmaceutical companies lost all those interests, most of the indications you would use FAAH inhibitors for would fall under psychiatric and neurological. And so the drug, there was just like no interest for years.
Hmm.
And ultimately, it was really the cannabis withdrawal study that was spearheaded by Cyril D'Souza that got the kind of was really the first data that was done which showed some kind of positive effect, where they showed that you had these effects on cannabis use, you had effects on sleep and anxiety, and so it gave some okay, maybe there's something here. And it was around the same time that Marcus and Leah had started their trial and on the
Hmm.
fear and stress stuff in humans, and now it seems to be ramping up. I mean, I know Johnson and Johnson's. I assume that they're pursuing the social anxiety angle more since their first trial was moderately successful. I know there's a lot of other companies now growing interest in FAAH inhibitors, given that there's now 4 published reports in humans in the last 2 years that have all shown significant effects, all relatively in the domain, at least every single study has had a measure relevant for anxiety or stress, and all of
Hmm.
them have showed benefit in there. So I think there is going to be clustering in that space. I would say in the next 5 years, we will have completed clinical trials for social anxiety disorder,
Hmm.
PTSD, probably other flavors of anxiety, generalized anxiety disorder, maybe phobias, maybe panic disorder.
Yeah, I was gonna say, like, based on everything I'm hearing, it sounds like this is a promising avenue, not just for something like severe PTSD, but almost any anxiety or stress disorder, even if it was mild.
Yeah, I mean, some of the stuff like that my lab's doing now, we're actually becoming very interested in this idea, kind of going back to what I'd said about how, I view anandamide and endocannabinoids as being homeostatic regulators. And in that context, we've always focused on how stress disturbs the system, and therefore stress messes up anandamide signaling, and that may result in changes in anxiety. But it made us start thinking about, there's a lot of other chronic disease states that have comorbid anxiety with them, and many of them are stressors on the body. So, we started looking at things like gut inflammation, so
Mm.
colitis. We just published a paper not long ago now looking at comorbid anxiety and colitis. And what we found was that the kind of persistent inflammation that exists when the gut is inflamed is enough to drive the HPA axis to get this persistent elevation in CORT, which recruits CRF signaling in the brain,
Mhm.
which seems to be able to cause FAAH activity to jack way up and anandamide levels to plummet. And so, and we also similarly did a thing using an epilepsy model showing the same thing, that sustained repeated seizures resulted in this kind of comorbid anxiety, which we know is very present in epilepsy in humans. And again, the FAAH inhibitors were very effective in reducing that, because it seems like there's this crash in anandamide from these chronic disease states, and that loss of anandamide might incidentally be resulting in the development of this anxiety that develops with the disease. I mean, it's interesting because you look at kind of some of these diseases, and you can give people with epilepsy anti-epileptics, and they target the hyperexcitability. But I mean, in many of these disease states, same with colitis, the primary treatments that they use don't address the comorbid psychiatric issues.
Mhm.
And that kind of anxiety and depression that hovers around a lot of these chronic disease states is a massive quality of life impediment that largely goes unaddressed. And we became really intrigued because Dave Finn over in Ireland has, kind of long been looking at these relationships between pain, inflammation, and cannabinoids. And he had a review a few years ago where they were looking at a lot of these studies that had looked at chronic pain, especially inflammatory pain, where cannabinoids have been used. And what they found was that even if it wasn't that effective as an analgesic, in almost every single study, it improved the comorbid psychiatric side effects. And in every study, very much people reported improvements in quality of life, even if the pain was still present. And so I've started to believe that these are dissociable phenomena that,
Hmm.
I mean, they are driven by a common genesis in some capacity, but I think once they fully manifest, they are targetable separately. And so I think there's a lot of value in looking at the cannabinoid system as a mediator of kind of comorbid psychiatric issues and a lot of chronic disease states. And in my personal belief is that's why so many of these chronic disease states, people self-medicate with cannabis
Hmm.
because it helps them regulate the anxiety and the sleep disturbances that are present. And even if it's not curing the disease, it makes day-to-day life more functional and better, and that's relevant. And I
Hmm.
think that that is actually a domain that has been largely sadly ignored from a pharmaceutical perspective because they focus exclusively on primary outcome measures.
Hmm.
I mean, you talk to people who suffer from a lot of chronic diseases, and it's how they can survive day to day. People are, I think most people aren't expecting a cure at this point for a lot of these things that we don't even really understand the biology of. But if you find a tool that helps you manage yourself despite having affliction from a disease, that's a huge benefit.
Yeah, and what about, so you mentioned colloides and comorbid anxiety, and the FAAH inhibitors might help with the comorbid anxiety. But if you're inhibiting FAAH and increasing anandamide levels, is that happening at all in the periphery? And are there any outright anti-inflammatory effects from elevated anandamide outside the brain?
Yeah, I mean, so we had done intentionally central administration because we wanted to see if we could manipulate the anxiety independent of the colitis,
Mm.
even though it actually did seem to improve the colitis a little bit too, which is
Hmm.
interesting. But yes, we know if for systemically administered FAAH inhibitors, like we know that there's been multiple studies that have shown that they can improve colonic inflammation as well. So the argument we made in the paper is, yeah, based on those studies and our data, this could in theory be a two-pronged approach. I mean, and you could certainly make the same argument for epilepsy because we know cannabinoids have the potential to curtail some of the hyperexcitability that exists in seizure disorders. So again, you might have some benefit in that capacity, as well as improving some of the comorbidity issues with anxiety and depression.
Well, you mentioned very briefly there that so you did studies where you're putting a FAAH inhibitor directly into the brain on purpose because you wanted to separate out the anxiety effects in the brain from the actual colloidus part of it. But you said you noticed some effects on the colloidus nonetheless, and this is not an area I know a lot about, but it's triggering some memories I have of reading about gut-brain interactions.
Yeah, yeah. I
And
mean, there's definitely something there. I mean, and others
yeah,
have previously shown that you can play with central cannabinoid receptors and actually have influence on peripheral inflammatory processes in the gut too. So it wasn't entirely surprising, but it did surprise us a little bit because ours was an acute administration,
hmm,
and the other ones had been more sustained. I mean, ours was a few hours, so there was the opportunity for something. The effect wasn't massive, but it did definitely bring down the inflammation slightly. So that was interesting, but yeah, the endocannabinoids definitely have a lot of this, you know, brain body. They influence a lot of these brain body perspectives. I mean, even the feeding stuff, there's a huge wealth of information outside of what we know how it influences hypothalamic circuits. There's a lot of evidence to suggest that cannabinoids peripherally can influence feeding states by regulating vagal nerve activity or regulating sympathetic outflow. Um, and they can regulate metabolic processes. So you can give drugs that don't enter the brain, um, and they still have impacts on feeding. I mean, there's this very fascinating story that has really not ever hit the light of day, from a drug company that had developed a peripheral CB1 agonist years ago. So they thought, if we make it so it can't enter the brain, they would be able to be devoid of psychoactivity.
hmm,
And since there was growing evidence that a lot of the analgesic effects of cannabinoids could be driven by peripheral mechanisms, that kind of peripheral nociceptors, that they thought, okay, we can make a peripheral CB1 agonist and use this as a means to treat pain without psychoactivity. And they presented the data at a cannabinoid meeting, and I don't think it's ever been presented anywhere else. The drug had to be canceled, I think, in a week. Every subject developed fatty liver and gained a significant amount of weight. Their ALT levels shot up. They had clear signs of liver inflammation, and they had observable fatty liver, and they had gained, I think, something like 4 kilos or something in 10 days.
oh wow.
Like they literally referred to it as the foie gras effect. They said they'd never seen anything like this and they had no way of explaining it. But they said the only thing they could come up with was that there must be some competing central process that cannabinoids activate that limit this from happening. And when you
Hmm.
exclusively target only peripheral CB1 receptors, you seem to really have this profound influence on peripheral metabolism and hepatic function that
W-
was like
Was there any
fascinating.
change in feeding, or was this all a metabolism thing?
There was definitely change in feeding.
Okay,
They
okay.
said that people ate a lot. So it was kind of an interesting thing. I mean, there was
Wow.
this, they even said like there was this thought afterwards when someone was asking them, and they said they'd have to see, they didn't know the species specificity of it because I don't think this happens in rats or mice. Like in none of the studies has anyone ever seen this effect come out
Hmm.
in rodents. But they were saying, this would be a very ethical approach to foie gras. That was like where the joke came from, because as opposed to force feeding animals, if you can actually induce fatty liver and promote food intake and very rapidly cause this metabolic shift, that would be a better approach to making foie gras than the horrific ways that it's kind of done right now in France.
So circling back to anxiety. So at the story in the beginning, this woman, she had pain processing issues, and she had basically a really good mood her entire life. She was almost unable to experience anxiety. We've talked about stress, we've talked about pain. Can you actually describe or define those terms and how they're different? And maybe
Stress and
let's start.
anxiety.
Yeah, stress, anxiety, and pain. Yeah.
Okay. I mean, I'm not a pain person, so I won't I mean, I kind of think of pain as like a noxious stimulus, a response to a noxious stimulus, like a burn or an injury in some capacity. I mean, there's no denying there's an emotional component to pain.
Hmm.
Like we know that like the degree of an injury and the personal, per like the way that an individual experiences pain don't correlate very well. And certainly in chronic pain states, you see this. So pain can certainly be influenced by stress and emotional state for sure. Um, so if we're talking about stress and anxiety, I think that like these two are definitely dissociable phenomenon. Stress is kind of harder to define because I feel like it's very nebulous. And like a joke, it was kind of like something that can be a noun and a verb or a fucking shit. I've forgotten the other
Or an adjective.
adjective. Yeah. Just the way people describe it. You can be stressed out. It's something that can happen to you. Stress can be a thing. So I think the easiest way to describe stress in the most basic terms that's all encompassing is that it's a real or perceived threat to an organism's well-being. So anything that would challenge the well-being of an organism, its survival, its ability to, reproduce, its ability to acquire food, its ability to do anything, a challenge that it would be presented with would be considered a stressor. So, and this can be very different from person to person. Some people will perceive things as very stressful that others find quite innocuous, and it's shaped by personal experience and history, things we've gone through. So I think stress is kind of a constellation of biological responses that happens in response to this challenge. So, like I said, you get an adrenaline response out of the sympathetic system. You get a glucocorticoid response out of the HPA axis. You have kind of behavioral changes that occur, and I think anxiety is one of the behavioral changes that tends to occur in tandem with stress. But I think stress and anxiety are dissociable from that. And what I mean is, anxiety is kind of a behavioral state where there's not an immediate present threat, but there's the sense that something could go wrong. So the way that, and people often confuse anxiety and fear, and the way that we always try and, I live in the Rocky Mountains, so the joke that we have up here is how you separate anxiety from fear is anxiety is going on a hike and seeing a sign at the bottom that says someone has seen a bobcat today. Fear is when you see that bobcat.
Hmm.
So there's a difference between the idea that there could be a threat in your environment, and that's what anxiety is, whereas fear is typically the response to a real and present threat. And so anxiety is kind of a state where a lot of our systems move into a state of vigilance, like I was saying before, where we're trying to perceive our environment more. We're trying to find where a potential threat could be so we can identify it, assess it, and then respond accordingly, whether it's actually a threat or not. So when we become stressed out, anxiety is one of the hallmark behavioral changes that happens with stress, because
Hmm.
obviously when a challenge presents itself, usually part of what we're trying to do is assess the nature of that threat that's presenting itself. So I would say the anxiety component is very typical to stress, but there are people who have chronic anxiety in the absence of any
Hmm.
observable stressors in their life. I mean, this is kind of the nature of anxiety disorders, and that's why I always think that anxiety is separated from stress in that sense, because it's really a behavioral state that can occur. And if the brain's filtration systems for how we take in environmental information and how we process it, if that's just skewed slightly, so that, something like we'd said before, like the prefrontal cortex isn't talking to the amygdala and helping to reference previous experience to understand safe versus threat. You might just get this development of an anxiety state in the absence of anything, because it's kind of become this default mode that will favor survival.
I see. So, stress, anxiety, fear, we've talked about FAAH inhibitors and how the endocannabinoid system ties into things like stress and anxiety. We talked about pharmaceutical FAAH inhibitors and the promise they show, but another FAAH inhibitor that is interesting that people know about but might not know is a FAAH inhibitor is CBD. So, can you talk a little bit about CBD? How good of a FAAH inhibitor is it? And what kind of doses do you think are needed to have that activity happen? Are people that are consuming 10 or 20 or 30 milligrams of CBD that they bought at the store likely going to see that kind of effect?
Well, the first thing I'll say is I'm not convinced CBD is a FAAH inhibitor. It
Ah,
may be a FAAH inhibitor. So CBD, the one thing I think that has been more consistent is that CBD has been found to elevate anandamide levels. So there has been some evidence to suggest that it inhibits FAAH, but there's also been evidence that has suggested that it actually triggers the biosynthetic enzyme responsible for anandamide. So it causes that enzyme to become more active. So that makes more anandamide. There's also evidence that it might block anandamide transport to metabolism and therefore keep anandamide around longer. And lastly, there is actually, because FAAH sits tethered to the endoplasmic reticulum, which is inside a neuron, like not near the membrane, it's deep inside. There is a binding protein that anandamide has to couple to move through the cytosol to go from the membrane into FAAH to get chewed up. And
mhm.
that's called fatty acid binding protein 5. And we know CBD binds to that and can displace anandamide from
I
that as
see.
well.
So
So there's actually
somehow,
4 separate mechanisms. Yeah.
okay.
So yeah, it does seem like CBD can elevate anandamide, maybe through FAAH, maybe not. But well, the first thing I'm going to say is I don't believe that anyone taking oral oils of CBD at 10, 20, or 30 milligrams is doing anything outside of creating, lack of sounding crude, very expensive shit. Because the bioavailability of it is so incredibly low. From what we've seen from the studies that come out of Johns Hopkins, you basically don't even get detectable CBD in the blood until you are consuming a dose of about 100 milligrams.
And is that from swallowed pill form CBD?
Uh, It's from, yeah, I mean, sometimes it's oil mixed into something that's swallowed and ingested. Sometimes it's in a pill form. It's not a sublingual. So, I mean, the pharmacokinetics in sublingual are definitely a little bit more unclear, I would say.
Mhm.
But from most of the PK work that's been, and even the stuff with Epidiolex from GW, the bioavailability is pretty poor. And one of the things that I've learned about CBD dosing in the general public is most people are not aware of what the clinical dosing actually is for CBD. I mean,
Mhm.
if you look at those pediatric epilepsy trials, those kids are getting, they refer to it in mgs per kg per day. And those kids are getting like 20 mgs per kg per day. That's per kilogram.
So
So, I mean,
how many raw milligrams would that be?
for some kids, I mean, this is like, because the dosing they use is 10 or 20, but the raw numbers I've seen go up to 2,500 milligrams.
Mhm.
They definitely are almost always over 1,000. I've never seen a pediatric epilepsy study really that's shown benefit where there's dosing per child that's come in under 1,000.
Mhm.
And
So as a rule of thumb, swallowing CBD at low doses probably isn't doing much. Is
yes,
that
I would say so. I would say it's something that I've actually tried to understand this, because if you go through the literature, I've never seen a clinical study that has used oral CBD at a dose lower than 300 milligrams that's ever seen a positive effect.
mhm.
And every failed trial of CBD I've ever seen is those that hover around 30 or 40 milligrams.
Do you think
So
it's possible that other routes of administration could allow for some efficacy to be observed just because the oral route is so inefficient?
So there are pluses and minuses to different routes of administration that you'd see with something with CBD. So if it goes pulmonary, so if you inhale through vaporization or smoking, the bioavailability is way higher. You're going
Mhm.
to get massively higher levels of CBD in the blood. The only downside to the inhalation route, outside of potential damage to the lungs from inhaling, is it's also cleared a lot faster. So what you end up getting is a spike. So you get a
Mm.
lot of CBD in the blood and then it goes away. The reason for the use of oral in the pediatric epilepsy studies is twofold. One, obviously they're not going to get kids to inhale or smoke anything, so
Hmm.
that had taken it down. But two, they are trying to achieve stable blood levels of CBD around the clock and dosing. And
Hmm.
basically, the approach they've taken is oral has terrible bioavailability, but its half-life is so much longer once it gets in the system because of the way that it slowly diffuses out of the gut.
Hmm.
And so they just overboard dose in that route so that they achieve the blood levels that they need, but they're stable and they're not spiking like this where they're going up and down. They kind of go up and they'll stay up at a more appropriate level. Um, so certainly an inhalation route would get much better bioavailability, but it's also much shorter lasting. So I
Hmm.
guess it's just a question of what someone is trying to go for. I've been baffled by, because I know physicians who tell their patients to take 10 or 20 milligrams oral CBD oil. And when I've asked them and said, like, you're actually a doctor, like you should be familiar with the clinical studies that have been done. So how do you reconcile, like, in good conscience, basically telling people to continue taking a placebo? Because that I can't honestly wrap my head around how, I mean, the way I refer to it is it's CBD homeopathy at that dose. Like there's an essence that may grace your body of CBD. If you take 10 or 20 milligrams, you're shitting it out. Most of it's not leaving your GI tract. So I really struggle with understanding how you could do this. And their response is, well, the patients tell us that they find benefit from the dose, so we don't want to tell them otherwise because they're benefiting from it. And I'm not necessarily against the benefit of placebos, although I have a little bit of an ethical issue when people are spending their own personal money on it as opposed to it
Hmm.
being something that's prescribed. I mean, Canadians, so I'm thinking of this differently, obviously, in terms of the medical system, but it's a little different if it's something that's prescribed and covered for you versus it's something that you're shelling out money out of your own pocket, especially if you don't have the financial ability. Because even 20 megs of CBD oil a day is not cheap. You're still spending a decent amount of
Hmm.
money on that accumulatively.
So, what about something like a sublingual absorption? Is there anything known there, or would you expect in principle that to be more bioavailable?
I would guess it'll be a bit more bioavailable. I mean, there are certain things with oral we know. Like if you take oral CBD, if you eat a fatty meal right before you take it, your
Hmm.
bioavailability goes up like fourfold. It
Okay.
dramatically increases. So I think if you take it on an empty stomach, you haven't eaten for hours, the prediction is that it's something like 4 to 6% bioavailability of what you take of CBD actually gets in your blood. And if you eat a fatty meal and take it, it goes up to like maybe 20%. So it does improve it a fair amount.
Hmm.
So you can do that. Like obviously, if you consumed a fatty meal and then took it, it would be better than not. I would imagine just sublingually, you'd probably get a bit more. I don't know. I mean, even in the oil studies, they always acknowledge that some of it probably is absorbing sublingually because it's going in the mouth, unless it's in an encapsulated pill. The oil goes in the mouth and some of it's going to get into the membranes there. So you probably are absorbing a bit of it. It's just not entirely clear.
Hmm.
So I would guess yes, but there's just, I mean, the whole idea of even doing kind of good clean pharmacokinetics on anything is there's so few people doing this. I mean, there's the group at Hopkins, which is really doing a good job of this, I would say, because they're giving us the most information comparing like oral, inhaled, vaporized versus smoked, CBD versus THC. So they're really kind of nailing down the bones of this. But everything aside from this has largely come from like GW for their Epidiolex
Hmm.
studies and some random clinical studies that have just measured blood levels.
Hmm. What do you make of, so just knowing what you know about the endocannabinoid system and clinical trials and how they normally function, typically when we develop drugs, it's a single molecule that has some level of specificity to it.
Hmm.
And they talk about the idea of a dirty drug. And people like Ethan Russo, who I talked to recently, are big on this idea of an entourage effect of combinations of drugs, even if some of them are present at very low levels that might not do anything detectable on their own, can actually change or enhance the therapeutic value of other compounds. Do you have any thoughts on that and how likely that is to be true for some of these cannabinoid compounds?
Well, I mean, one thing we know about CBD is it does, especially at high doses, it can saturate a lot of the enzymes responsible for breaking down other drugs.
Mm.
In theory, THC could too, but THC never reaches the levels
Hmm.
because of the intoxicating effect at relatively low doses. So people would never often achieve those levels, which is why we don't see a huge amount of drug-drug interactions with THC. But for example, the pediatric epilepsy studies, they have clearly found that the kids on the high dose CBD, they start seeing some of the other drug levels accumulate and go up because you're inhibiting metabolism. So I think if you're taking reasonable levels of CBD, there's always that concern. There was even a study that just came out recently that said dosing at like 1,000 mgs of CBD, which is not unheard of because that's the clinical levels. In healthy controls, they were able to see elevations in liver enzymes like ALT and stuff, suggesting that there might be some possible effects on liver. So I would assume that you have probably a pharmacokinetic interaction that could occur with other drugs, independent of anything else. In terms of kind of the dirty drug, like this I mean, do you mean like entourage effect kind of stuff like you've been
Yeah,
talking?
exactly.
So I think that there's probably something to an entourage effect. I don't know if we're going to be able to chase it down, to be honest. And I say
Mhm.
that because I think it's going to be much more complicated. Like the idea of trying to understand singular substances and then multiple interacting substances and how they're going to interact with each other is very tricky. I mean, I know the group in Australia has been screening a lot of the kind of lesser cannabinoids and other things that are in cannabis, like,
Mhm.
outside of CBD and CBG, some of the terpenes and stuff. And at least what they're looking at is they're trying to see interactions at CB1, and they're not
Yeah.
seeing that it's interacting there. But that doesn't mean that
No,
these,
yeah. I
yeah,
mean,
it could
that's
be.
a weird experiment because I mean, as far as I'm aware, none of those other compounds are known, other than THC to really do much at CB1 anyway.
Mhm.
So you can it's sort of really hard to definitively prove a negative in an assay like that. But to your
Yeah.
point, it's also really hard to prove the positive statements here. And the only way I can even think about it is you would have to give humans or animals in some way that was rigorous an entourage of compounds and give some other group a different entourage, and you'd have to measure the effects at like the behavioral level and the neurological level.
I mean, there are groups starting this. I know Ryan Vandria and Ziva Cooper, like
Yeah.
Ryan's at Hopkins and Ziva's out at UCLA. I mean, I know they're starting to look at, I mean, their approach is they'll take like THC and then add a single terpene or a
Hmm.
single
Hmm.
cannabinoid and then measure, like what you're saying, whatever their endpoint is, be it pain responses or your own psycho or your own, like, how high am I, that kind of thing. Is this rewarding? Is it aversive? So they're kind of doing some baseline looking at that to see. I think that's probably the best approach because that will at least help narrow down
Yeah.
which molecules might be more interesting for entourage type interactions. And then you can start trying to understand the molecular aspect of that a little bit more in a different capacity. So I think there is potential for this to go down. I agree. The reductionist approach looking at CB1 is, I mean, I think it's necessary to rule that out, but I don't think that that argues against an entourage effect because all that tells you is, yeah, molecules we didn't think were acting at CB1 indeed are not acting at CB1. And so we're not seeing an interaction at that point. But, because cannabinoids, THC is going to affect, neurotransmission and synaptic transmitter release. If you have something that indiscriminately affects the GABA system,
Right.
independent of CB1, that obviously could interact with how cannabinoids are influencing GABA transmission. So
Hmm.
I do believe that there's potential for this. I mean, even if you take something like CBD, whose pharmacology, like, what is CBD doing? No one really knows. Sure, maybe it's boosting anandamide in some kind of ill-defined way. I mean, the other mechanisms are maybe it's kind of like an allosteric modulator of serotonin 1A receptors. That's possible, although there is some arguments against it as well because people have had some problems replicating it. CC Hillard's put together a really nice mechanism, I think, by showing that CBD actually inhibits adenosine uptake because it competes for the adenosine transporter essentially. And so you'll enhance the synaptic actions of adenosine with
What
CBD.
is? Can you describe what adenosine is?
So the way I tend to describe this, if I'm talking to people in the general public, is adenosine as a system is actually quite similar to endocannabinoids, but it is what is inhibited by caffeine. So, like, if you want to boil it down to it, I mean, adenosine functions similar to the endocannabinoids, how it regulates transmitter release. But so caffeine inhibits adenosine, and that's part of the mechanism of how caffeine, causes alertness and affects arousal states. So if you think of CBD, I always think if you're going to find a drug that you want to relate it to based on that mechanism, I would say it's almost like an anti-caffeine. And
Hmm.
that would explain some of the kind of, supposed, pro-sleeping somnogenic effects and some of the anti-inflammatory effects and some of the other mechanisms of CBD certainly make sense in the context of enhanced adenosine signaling. Um, but who knows if that's going to be kind of the be all and end all. I think this is the problem when you have a drug that, like, you know tickles so many neurochemical systems but doesn't really get into bed with any of them. So
Hmm. Yeah.
it's
So it's doing a lot, which gives it a lot of potential, but it's difficult to actually work out what's doing what.
And I would say that one of the biggest disconnects that a lot of people in the general public don't understand about why the medical and scientific community is so reluctant about CBD is, um, I would say general scientists become skeptical of massive claims when there's no clear mechanism of action. Like when you cannot, I mean, I know like Brian Roth screened CBD through his G protein assays and said he couldn't see it binding to or influencing a single receptor.
Hmm.
And that included serotonin 1A. But he looked through every GPCR in his library and couldn't see CBD interacting with any of them. And he said maybe it was the dosing wasn't high enough. But I mean, the reality is the people are claiming effects from CBD at doses that are like incredibly low. So if it was actually doing something on a biological system, I feel like we would be picking it up at some point. It's hard because the problem is cannabinoids are so sensitive to
Unless
expectancy bias.
I guess
But
the potential counter argument there would be, earlier you cited this, this other type of protein interaction CBD has. So maybe it's not having strong effects at a receptor level, but maybe it's some other intermediary that it's influencing.
oh, it's certainly possible. I think the thing is at this point, the amount of people who've been studying it, no one's been able to come up with a consistent readout or story that makes sense.
Hmm.
And I think that's been one of the things that's hampering it. And I think the problem is, unlike the kind of history of cannabis, because the history of cannabis influenced research in the cannabinoid system so much. We know people have been using it for centuries to regulate pain. We knew that the munchies was a really big side effect associated with cannabis use. We knew that people used it to reduce stress and anxiety. So it really dictated the physiological processes that became investigated, and it made sense. So we had insight. We knew to look at pain circuits. We knew to look at feeding circuits and emotion circuits because there was these very prototypical effects of THC that had been well established. CBD, on the other hand, 15 years ago, I could have walked into a room of 1,000 people and said, how many of you know what CBD is? And maybe 3 would have raised their hands. Now, like 998 of them will. And the
Hmm.
problem is in that period of time, the amount of anecdotal reports that have come out, I would say it would be easier to assemble a list of diseases that people claim CBD doesn't cure than things that people claim it does, because virtually every single disease out there, someone has made some argument for how CBD cured it for them. And so, from a scientific perspective, like even how I was referencing that anecdote stuff before, we can't believe any of it because it's just gotten to a level of such nonsense where everyone believes that every single disease is cured by a molecule that doesn't have clear pharmacological activity and is being taken at doses that doesn't even have clear bioavailability. So it's very hard to take insight from the clinical perspective and the human anecdotal response in this and try and design experiments to test things out of it. And so I think a lot of it has just become this like Me Too redundancy thing where people are like, well, let's look at things that were already affected by THC, like pain and inflammation and sleep and anxiety,
Hmm.
and see what CBD does. And I'm just kind of like, sure, but like if we're trying to reference cannabis as the reason we look at these, then why would CBD be relevant? Because CBD and THC compete for biosynthesis. So given that for the last 30 years, THC has been so enriched in cannabis breeding
Hmm.
because that's what people want out of it, by default, CBD has been bred out of cannabis almost
Hmm.
entirely. So over the last 20, 30 years, if you took a strain of street cannabis, you would be lucky to have found 1% THC or CBD in any of it.
Yeah, I can tell you. So we're putting together a paper right now that looks at just under 100,000 laboratory testing samples of cannabis flower, and conservatively, I would say more than 95% of cannabis flower used commercially has less than 1% CBD.
Yeah, that's about what I would predict. So that's consistent with it. So this idea that's permeated the public that CBD is medical and THC is recreational makes zero sense to me, because I'm like, I don't understand how in the same breath we can be referencing the fact that people have been using cannabis in a medical context for decades to centuries, depending on the culture, and then yet at the same time try claiming that this is not due to THC's actions, where we know it has a pharmacological effect on CB1 receptors, and we know that CB1 receptors regulate the processes that people claim are being benefited by it. We have no concept of this with CBD. So
Yeah, I agree that, one of the biggest misconceptions that's out there is this THC is the fun one and CBD is the medical one. Is there still, historically, I think there's been, quite an allergy to THC in at least certain parts of the clinical community, where most of the studies have looked at, the abuse potential and things like this. Do you see that changing? Is there a lot more clinical work happening these days around THC and its potential medical benefits?
um, I mean, the funny thing is, like, everything that had been done clinically had almost all been done with THC private, because it had all been done with nabilone or marinol or isolated THC. And all
Mhm.
these medical studies that had been done, like everything that had been done with Sativex, that was mostly THC. Everything that had been done in Canada was with nabilone. So it's weird to me. Like, I'll talk to clinicians and they'll be like, well, there's no evidence at all that cannabis has any benefit in PTSD, and I don't see it doing anything but damage. And I'm like, it's really weird that you say that, because like there is clear data that isolated THC has been done in experimental settings. And granted, there are some issues with the study, but you can't deny that there's data that exists. And to
Mhm.
say that something that was found with isolated THC will not generalize to cannabis, sure, that's possibly true, but that's incredibly naive.
It's weird, yeah, because it is mostly THC.
Yeah. And so to me, it's very strange. And I think, to be honest, I mean, and I'm not a physician, so I'm not, I always tell people I'm not bound in the same problematic space that a physician could be,
Mhm.
because I'm not prescribing something. I'm not telling people what to use. I'm just looking at what the data is and repeating it. And so it's different from a physician's perspective, because I feel like, especially on the heels of the opiate crisis that's been going on for years now,
Mhm.
physicians are incredibly reluctant to say anything that could be interpreted as endorsing a drug that could have some abuse potential,
Mhm.
because of the history of what's happened in the context of prescribing drugs that have abuse potential.
Mhm.
And will that change? I don't know. I mean, we're over 2 years into legalization in Canada, and now, I mean, two in some. And the data that's been put out from Health Canada has generally shown that there's been zero impacts on almost anything that we can measure in the country. Like, it's like Y2K all over again. It's the most anticlimactic. I mean, and the thing that blows my mind is I'm fundamentally confused why more countries aren't talking about how boring legalization in Canada ended up being.
Mhm.
Like, it didn't do anything. And so I'm surprised that in the space of that time that more physicians haven't. I don't know. I've tried to have conversations with physicians about it. I mean, there's clearly risks associated with cannabis, and it's not something that works for everyone. And there's clearly subsets of people that respond badly to it. And it's easy to say that, though. It's
Mhm.
just, I feel like there's this insane push to always believe data suggesting adversity, and there's this very strange reluctance to ever embrace any data that suggests benefit.
Mhm. You mentioned opioids briefly. I'm going to talk to Yasmin Hurd in the near future, but setting aside the question marks around mechanisms of action, what do you think about the work that suggests CBD might be used as an addiction treatment?
I mean, Yasmin's work's probably some of the most interesting that's been done out there, because it does. I mean, I also like how Yasmin went pipeline from animal studies into humans based on a phenomenon she observed there. I think I mean, to me, it makes sense in terms of the measures she's looking at, because she's largely looking at craving and anxiety measures
Mhm.
in individuals with opiate use issues. And so I'll be curious to see what Yasmin's data looks like in the larger context of a relapse study, where they're actually looking at more direct measures of opiate use
Mhm.
and outcome measures. But I, I mean, I think it's an interesting approach that she's taking.
Can you just briefly summarize for people, what's the basic summary of that work?
I mean, it's been a while since I've read it. If I can remember it correctly, Yasmin was looking at individuals who had opiate use disorder, and were abstinent, and they gave them CBD and looked at measures of, I think it was 300 and 600 milligrams in her studies, and I think both of them had some efficacy. And I think I can't remember the duration of dosing, but I know that the measures they saw signaling were it reduced some scores on craving and it reduced signs of anxiety. And the reason those are relevant is because those obviously tend to be big predictors of relapse.
Mhm.
Because if someone is, we know stressors and anxiety can be big triggers for people to relapse drug use. And we know that craving obviously has a huge component in relapse. So if you have a pharmacological intervention that can reduce some anxiety and also reduce craving, by logic, you would assume that that would have benefit in actually curbing use long term, is why it'll be interesting to see when that gets put into test in an actual clinical study, if it actually shows that it has efficacy in that space.
Mhm. So in our last few minutes here, what's on the horizon that's interesting, either in your lab or generally in this field of endocannabinoid biology?
I mean, I think despite the fact that my career has almost entirely been endocannabinoids and not much in cannabis, we've recently moved into some cannabis stuff. And I think the thing that's become interesting and why this is cool is been with this kind of repurposing of these vapor chamber technologies that was developed for nicotine delivery through
Mm,
vaporization. So there's Maury Cole has this company down in San Diego that makes these vapor chambers, and he'd worked with a lot of the nicotine researchers, and some of them started moving into cannabis, like Mike Taff, then at UCSD, and he started working with Maury to convert these chambers so you actually basically try to replicate like vape pens, where you take cannabis oils or THC distillates and you vape them to animals. And this may not sound like a huge technological advancement, but the entire field of understanding the effects of THC on the brain has almost entirely been performed using injection approaches for the last 30, 40 odd years.
meaning injecting THC
Yeah.
directly into the brain of an animal.
Or into the periphery in a big bolus where you dump it in.
Hmm.
Now, what had not been appreciated that much is how different the pharmacokinetics of it are. So what we've learned, we have a paper right now where we've just compared inhalation to injection, and we've looked at the pharmacokinetics of it, and others have started to do this too. And what we've seen is very clear. If you inject THC, it's a lipid. A lot of it immediately sequesters into the fat. This was work Steve Mahler and Daniele Piamelli did down at Irvine to show that you actually get a huge amount of THC that goes right into the fat pads. And what seems to happen then is that THC then slowly leaks out into the
Mm,
bloodstream for hours. And because it's leaking into the bloodstream before it hits the brain, it often hits the liver, which means it makes a whole boatload of this metabolite called 11 hydroxy THC, which is actually a little bit more potent than THC itself and has more readily access to the brain. And so what you see from an injection is if you measured a single blood time point of blood THC levels, someone would say, oh, it looks like roughly the blood level someone achieves from smoking a joint.
mm,
But what they don't realize is when you smoke a joint, that's the peak level that lasts for like 5, 10 minutes and then it clears. And when you inject it, it sits there for like 5, 6 hours. And so from a pharmacological perspective, what this means is that injections of THC are causing hours of sustained activation of CB1 receptors that is not at all reflective of what's happening in reality when someone
mhm,
inhales cannabis. The other problem is a lot of the animal work had been using synthetic cannabinoids that weren't THC just because of FDA or DEA issues with it being a Schedule 1 drug.
mhm,
And they're like, oh, it's easier and it's pretty much the same thing. And now we know that's not true because we know the synthetics are biased agonists and recruit all these different pathways. So basically, we had kind of looked back on this whole literature of animal studies looking at what people were assuming cannabis was doing to the brain is being based on injection studies of THC or synthetic agonists, which don't behave the same. And so this idea of using a vaporization approach so animals inhale it, while it again doesn't sound like that technological, when you think of things like optogenetics or dreads or stuff, it's not that technological advancement, but it is a huge step forward in the field because now all of a sudden we have an approach that really models how people consume it. And it models the PK of how THC is going to be
mhm.
metabolized in the body and how it's going to impact the brain. And we are definitely seeing different effects from doing this. And I think that now this is becoming the new wave. There are multiple labs in Canada using this technology and tons
Mhm.
in the States.
Can you just describe very concretely like what the setup is that a mouse or a rat would be in and what they might be doing after inhaling?
So the way these chambers work is you can either, like we have two setups. Some of them you just put the animals in kind of a clear plexiglass chamber, and sometimes we have a big setup where we can just put their whole home cage in and leave them so we don't have to disturb them.
Mhm,
The cages are the boxes they're in then become sealed. It's kind of a negative pressure environment, and it has like basically a vape chamber that's pretty similar to what you'd find in a pen, but a bigger one that you put the oil into. And using this system, you can kind of tweak it using the computer setup to do controlled delivery. So we do a 10-second puff where it basically blows THC vapor into the chamber for 10 solid seconds. You get like about 30 seconds of a Cheech and Chong room that's just like cloud and you can't really see anything. And
mhm,
then through the negative pressure, it slowly dissipates over the next couple of minutes. So the animal inhales it during that period. And we do that for 10, 15 minutes. And that gets us at the blood level we would expect from someone smoking a joint or two.
mhm,
And the animal's clearly intoxicated. Like you can see it, like they kind of sit there, they won't do a lot, their eyes are kind of closed. We've measured body temperature, they get a drop in body temperature. We've started doing a lot of munchies studies, and they clearly definitely eat more. They go into for 30 minutes after they're high, they binge. But what's interesting is they binge and then they don't eat for like 4 more hours.
Oh,
So it's
wow.
like while they're high, they just and we can pre-satiate them and then they'll just keep eating beyond satiation.
So
But
it's just like humans.
yeah. And then as soon as but the interesting thing is like, so pot smokers in humans is not associated with obesity. In fact, pot
Right.
smokers more often than not are less obese than the general population. So it's interesting because what you end up seeing, what we keep seeing in the animal stuff is this binging that happens in a very restricted time window and then like almost no food consumption for the period afterwards. We haven't like got them successively high to see if we can get feeding bouts to occur over
Hmm.
and over again, but we definitely get a clear munchies phenotype out of them.
Is
And
there,
then
I assume someone is measuring motivational drive. Like if you train the animals to do a task, are they just lazier after?
well, so we had previously done some stuff on THC injections with that. We're trying to do this more now, looking at reward learning tasks and how it influenced that, as well as aversive learning tasks. My buddy Ryan McLaughlin, who's down in Washington, he was one of the other people that really kind of pioneered this. And he actually did the first thing. He was the first person that actually showed you could train animals to self-administer cannabis vapor hits.
Hmm.
So he put them in kind of basically an instrumental conditioning situation where the animals would learn to like lever press. And it's fascinating to watch because you could see the videos where the animals would like lever press and then run over to the port and kind of like bask in the vapor
Hmm.
hit as it comes. So they clearly enjoy it. It clearly is rewarding. They'll work for it. You know, you start doing extinction, you get extinction bursts where the animal like starts going crazy to try and get cannabis vapor clouds. So and it's interesting because then one of the things Ryan showed since then, which is relevant for me, and we found interesting as well, is that if you let animals self-administer cannabis and then you stress them out, they actually do have blunted stress responses, same as
Hmm.
what you see in humans.
Yeah.
So again, it's nice because we're able to recapitulate these kind of real life phenomenon that we see in cannabis users. And the reason I like this is I feel like now we can actually properly study cannabis. And I honestly feel like the way that the field has approached studying cannabis, again, largely due to this DEA regulations and the fact that this technology just wasn't easily accessible, has been subpar for some time. And I think it's really significantly influenced a lot of the way that we think, especially about the adverse effects of THC or cannabis, because a lot of these studies have been done with incredibly massive doses
Hmm.
that are having far more biological impacts on the brain and body than what we would normally see from cannabis consumption. And
Hmm.
so I don't doubt that there will be like adverse outcomes that are coming from this. I mean, we're starting to do some prenatal and adolescent exposure, and there's certainly things that you start to see. I mean, the prenatal stuff is consistent. You always see, reduced birth weight. There's consistently some behavioral phenotypes that kick in later in life. So there are effects that are clear that are coming out of this. So we're like, okay, we are confident that this is probably more reflective in a translational manner of what's really going on in humans than what we would have seen before that. So I have to say, I think this is actually a really exciting kind of new avenue of things. And it's a little bit out of my normal wheelhouse, but it's still kind of in the cannabis cannabinoid space. So
That's awesome. Yeah. So in the next year or two, we'll actually start to see probably quite a few animal studies come out that are using these paradigms.
yeah. I mean, there's at least 20 labs that I know of now in Canada and the States that have got this technology or variants of this technology working in their own groups and are doing studies like this. And I believe even Yasmin's moved over to the chamber approach as well now. I think it's just kind of like if you really want to be asking questions that are going to be relevant to humans, like the argument I always make is I'm like, we don't inject alcohol anymore. We don't inject nicotine. There
Yeah.
are other
Yeah.
ways that these drugs are administered because we as a field have recognized that route of administration impacts PK much more than people appreciate it. For cannabinoids, it's far more relevant than I feel like a lot of people had given credit for in the
Yep.
past.
Well, that's great. It's a positive note to end on. I look forward to seeing some of that research come out. Thank you, Matt, for your time. Is there anything, any final words you want to leave people with? Do you have a website or any place where people can find you and your latest thoughts?
No, I mean, I'll use this as a shameless pitch for an organization that I'm the director of, which is called the CCIC. It's an organization of physicians and scientists that kind of tries to promote accurate education about research and science regarding cannabis and cannabinoids, all the way from plant science, basic biology, through to human use, clinical outcomes, and public health. And we have an annual conference that's this year will be run virtually, which details can be found at ccic.net. But it's a great review of the most up-to-date science being done by Canadian and American cannabis scientists. So this year it's people like Ziva Cooper and Ryan Vandry and CC Hillard and a bunch of Canadian people looking at some of the data that's come out of Health Canada. We'll be, updating legalization data on use patterns and things like that. So I encourage people to look at the website and attend the conference if you can, if you're really interested in understanding more about kind of the current state of cannabis science in Canada and the States.
All right. Well, thank you, Matt.
Perfect.